Right now, as you are sitting there listening to this, your brain is quietly burning enough electricity to power a small LED bulb.
Which, I mean, that might not sound like a lot at first.
No, it doesn't. Until you realize that this quiet electrical hum is happening constantly, whether you're wrestling with a complex math problem or folding a mountain of laundry or just, staring blankly out a window.
Right, it never stops. It's relentless.
Exactly. You are casually carrying around the absolute most complex object in the known universe sitting right behind your eyes, and it is relentlessly hungry.
And that hunger is exactly what we're going to get into today.
Yeah, so today our mission for this deep dive is to completely demystify this astonishing structure for you. We're going to take our time, assume absolutely zero prior science background, and just strip away all that intimidating jargon.
Because the jargon really gets in the way of the sheer awe of it.
Right. It really does. We just want to explore the physical reality of the human brain. And exploring that physical reality requires us to immediately abandon, well, a lot of our preconceived notions about what a powerful machine actually looks like.
Oh, totally. Because when you look at the raw physical statistics of the human brain, they almost seem like a biological joke.
A joke? How so?
Well, we're talking about roughly 1.5 kilograms of soft tissue. It essentially has the consistency of firm jelly.
Firm jelly. That's kind of unsettling to think about.
Right. If you were to hold a brain in your hands, it would barely hold its own shape without the support of the skull and that cerebrocanal fluid it floats in. It's incredibly delicate.
But there's a huge paradox here, right?
The ultimate paradox.
Yeah.
That delicate, firm jelly makes up only about 2% of your total body mass.
Just 2%.
2%. But it demands and consumes a staggering 20% of your body's entire energy supply every single day.
That is just a staggering disproportion. I mean, imagine having this massive, sprawling house, and in the front hallway, you have one single, tiny, unassuming smart bulb.
Okay, I like this analogy.
Right, so you have this little bulb, but when the electricity bill comes at the end of the month, you find out that this one little bulb is responsible for a fifth of your entire power consumption for the whole house.
You would definitely think there was a short circuit.
Yeah, or you demand to know what on earth that bulb is actually doing to draw so much power.
And that right there is the ultimate biological mystery. Because if we frame this through the lens of evolutionary biology, we know that evolution is an incredibly ruthless accountant.
It doesn't tolerate waste.
Absolutely uncompromising. It
Right.
does not tolerate waste, especially not energy waste. Historically, energy meant finding calories in a very dangerous, highly competitive environment.
So you couldn't just waste it on something that wasn't pulling its weight.
Exactly. Evolution would never, ever maintain an organ that is this wildly metabolically expensive if it weren't doing something extraordinary to ensure our survival.
Yeah.
The energy cost is astronomical.
Yet, despite paying that caloric tax every second of every day of our lives, humanity as a whole understands this jelly less well than we understand, like the life cycles of stars burning billions of light years away.
It's true. We can map the cosmic microwave background of the universe, but our own internal wiring remains this massive uncharted frontier.
So to understand how this energy-hungry jelly actually works, we have to start by zooming way, way in. We can't just look at the whole 1.5 kilogram mass at once.
Right, you have to look at the microworkers.
The fundamental building blocks doing all the heavy lifting. We're talking about the neurons. And right off the bat, there is a massive historical correction that we need to address about just how many of these cells we actually possess.
Yes. This is a big one. For a very long time, if you opened almost any biology textbook or read a popular science article, you'd see the number 100 billion repeated over and over again.
100 billion neurons.
Right. It became this universally accepted fact. It was repeated so often that literally no one questioned it.
But it was wrong.
It was, in reality, just a very rough, rounded estimate. It wasn't until a highly careful, rigorous count was conducted in 2009 using a completely new method where they basically dissolved brain tissue into a uniform suspension, like a soup, to accurately count the nuclei, that the number was formally revised downward.
Okay, so what's the actual number?
The actual figure is roughly 86 billion neurons.
Okay, I have to stop you there because my immediate instinct is to look at those two numbers, 86 billion versus 100 billion, and wonder why a 14 billion difference actually matters.
It sounds like splitting hairs at that scale.
Exactly. When you're dealing with numbers that astronomical, isn't it all just essentially billions and billions? Does it really change our fundamental understanding?
It changes it profoundly. Because in science, precision is everything. 14 billion is not just a rounding error that we can just sort of wave away.
Why not?
To put that 14 billion difference into physical perspective, 14 billion neurons is roughly the entire brain capacity of a baboon.
Wait, really? A whole baboon?
A whole baboon. So for decades, we were casually tossing an entire extra primate brain into our estimates of human computation.
Wow. We were literally hiding a primate in the math.
Exactly. And the reason this matters so much isn't just about, getting the trivia right. It highlights how much of foundational brain science has historically been built on casual assumptions.
Like, we just accepted a nice round number because it looked clean on a chalkboard.
Exactly. It's a stark reminder that we need rigorous checking in this field.
That is a phenomenal perspective. So we have these 86 billion neurons, but they aren't even sitting in there alone, right? There is another type of cell that makes up roughly the other half of the brain's mass.
And they rarely get the spotlight.
Right. We have a comparable number of these. Roughly another 86 billion called glial cells.
Glial cells are truly the unsung heroes of the nervous system. And historically, there was a massive paradigm shift in how we understood them, too.
I read that the name itself, glia, actually comes from the Greek word for glue.
It does. And for decades, neuroscientists dismissed them as mere structural support tissue.
Like they were just holding the important stuff together.
Right. The prevailing thought was that they were essentially biological bubble wrap. They existed purely to keep the important electrically active neurons physically insulated and locked into place.
Just like packing peanuts for the brain, entirely passive.
That was the assumption. But today, that view has been completely overturned.
What do they actually do then?
We now understand that glial cells are incredibly dynamic, active players in the brain's environment. They don't just sit there. They are actively involved in chemical signaling.
Oh, so they communicate too.
Yes, and we manage the brain's highly specialized immune function, hunting down pathogens and clearing away cellular debris.
That's a lot more than bubble wrap.
And crucially, they actively regulate the connections between neurons, determining which pathways get strengthened and which get pruned away. The brain simply could not function without them. They aren't the scaffolding. They are an integral part of the active machinery.
So we have this unimaginably dense, active forest of neurons and glial cells. Let's look closer at the main characters, the neurons themselves.
Good idea.
The anatomy of a neuron can be broken down into three main functional parts, and it's actually a beautifully intuitive structure to visualize. First, you have the dendrites.
Right, the receivers.
Yeah, these are highly branched structures that radiate out to receive signals from other neurons. I sort of picture them like the sprawling, intricate branches of a mature oak tree, just reaching out in every direction, waiting to catch incoming signals.
That is an excellent visualization. They are the chemical and electrical receiver, the input zone, basically.
Then moving down from those branching receivers, you have the cell body, also known as the soma.
Right, the soma.
And this is the central hub of the cell, where the nucleus lives. It acts as the integrator. It takes all those thousands of inputs caught by the dendrite branches and makes sense of the noise.
It does all the math.
Exactly. And finally, you have the axon. This is a single long cable that carries the cell's output signal away to its next destination.
And the axon brings us to one of the most counterintuitive physical facts about the human body.
What is?
Well, when we think of a cell, we are conditioned to think of something strictly microscopic. We picture something we need a powerful lens to see.
Right, like an amoeba or a red blood cell.
Exactly. And the cell body of a neuron is indeed microscopic, but that single output cable, the axon, some of those single cellular extensions run the better part of a meter in length.
Wait, I really want to make sure I'm visualizing this correctly. You were saying a single microscopic cell body has an attached cable that is like 3 feet long.
That is exactly right. There are motor neurons where the microscopic cell body sits safely tucked inside the base of your spinal cord, but its axon stretches all the way down your leg, terminating at the muscles in your big toe.
That is wild.
It's a single, continuous, unbroken cellular tube. To put the scale into everyday terms, if you were to scale the microscopic cell body up to the size of a standard tennis ball, that single axon would be a garden hose stretching for over a mile.
A mile-long garden hose. That completely defies our normal sense of scale.
It really does.
And it immediately raises a huge mechanical problem in my mind. We have this physical hardware. We have the 86 billion neurons, the tree branches receiving, the cell body processing, and these mile-long biological hoses sending outputs.
Right.
But how does a signal actually travel across those massive distances without just, fading into nothing? How do they talk to each other? The brain, as we know, uses a bilingual system, right?
Yes, it communicates using both electrical and chemical languages.
So how does the electrical part work?
The distinction between those two languages is vital to understanding everything the brain does. So let's start with what happens inside a single neuron. Within the neuron itself, traveling from the cell body all the way down that incredibly long axon cable, the communication is purely electrical.
Purely electrical.
Yes. It is a wave of voltage change that sweeps along the membrane. In neuroscience, this event is called an action potential.
Now, I want to clarify how this electrical wave actually works because it's not like electricity flowing through a copper wire in a wall, right? It follows a very specific rule.
It does. It's an all-or-nothing event.
Right, all or nothing. You often hear it compared to a text message like, you can't half send a text, it either sends or it doesn't. But physically, I think a better way to visualize the action potential is like a stadium wave of falling dominoes.
A line of dominoes is a brilliant way to picture it because it perfectly captures the mechanism of how the signal actually propagates.
Right, because when you tip the first domino, it falls and hits the second, which hits the third, all the way down the line. You can't have a weak domino fall.
Exactly.
Once the first one tips past a certain angle, the entire line is going down with the exact same momentum. The wave travels all the way to the end of the axon without losing any strength because each segment of the axon is actively generating the next step of the wave.
And that domino analogy brings us right back to our opening paradox about the brain's massive 20% energy consumption.
Oh, how so?
Well, think about a mile-long line of dominoes. Once they have all fallen down, they cannot carry another signal until someone painstakingly walks down the entire line and stands every single domino back up, one by one.
Oh, wow. I didn't think about the reset.
Right. In the neuron, this standing the dominoes back up involves molecular pumps that physically push sodium and potassium ions back across the cell membrane against their natural gradients.
And I'm guessing that takes a lot of energy.
Immense amounts of biological energy. That is where a massive chunk of your calories goes. Just constantly resetting the electrical dominoes so you are ready to think your next thought.
That makes so much sense. But wait, if the electrical wave is a falling domino, if the signal strength is always exactly the same, an all-or-nothing pulse, how does my brain know the difference between, say, a gentle breeze brushing against my arm and someone violently slamming a door on my finger?
That's a great question.
Because if the single pulse is identical, where is the information about intensity coming from?
Well, if the information cannot be carried in the strength or the size of the electrical signal, it must be encoded in the timing and the frequency.
The frequency.
Yes. The brain encodes intensity through rhythm. A gentle breeze might cause that sensory neuron to fire an action potential, to knock down its line of dominoes maybe 5 times a second. Pop, pop, pop.
Okay, kind of a slow rhythm.
Right. But a slammed finger will cause that exact same neuron to frantically fire 50 or 100 times a second. Pop, 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 pop. The individual electrical spikes are identical in size, but the rapid-fire frequency instantly tells the central nervous system that this is an intense, urgent stimulus.
So the frequency is the message. But that electrical domino wave only gets you so far, right? It only gets you to the very end of the axon.
Exactly. And here is the twist that makes the brain bilingual. The axons do not actually touch their intended targets.
They don't touch it all.
No, they do not. And this is a foundational concept. At the very end of that long cable, there is a microscopic physical gap between the sending cell and the receiving cell.
A physical break.
Yes. It is incredibly small, only nanometers wide, but it is an absolute physical break in the circuit. This gap is called a synapse.
So the electrical stadium wave hits a literal cliff. It reaches the end of the axon and has nowhere to go.
Right. And electricity in the biological environment of the brain cannot simply arc across empty space like a spark plug in a car engine. The electrical signal hits a dead end.
So how does it cross?
To bridge that gap, the electrical signal forces a profound transformation. It triggers a chemical process.
Ah, here's the second language.
Exactly. When the action potential arrives at the end of the axon, the change in voltage forces tiny cellular packages to fuse with the membrane and dump millions of molecules into the gap. These molecules are called neurotransmitters.
Okay, these are the chemicals we hear about in popular culture all the time, right? Like glutamate, GABA, dopamine, and serotonin.
Those are the ones.
These are the chemical messengers. I kind of picture them as...
Broadly speaking, they fall into two camps. Some neurotransmitters, like glutamate, are excitatory. When they cross the gap and bind to the receiving cell, they essentially vote yes. They bring the receiving cell closer to firing its own action potential.
And the other camp?
Others, like GABA, are inhibitory. When they bind, they vote no. They actively suppress the receiving cell, making it much harder for it to fire.
So it's not a relay race at all. It's an insanely complex summation game. It's like a microscopic voting system happening in real time.
It is exactly a voting system.
Right.
Remember how we said the dendrites are like sprawling tree branches?
Yeah.
A single neuron's branches might be covered in 10,000 individual synapses. It is receiving chemical inputs from 10,000 other cells simultaneously.
That is just mind-boggling.
It is. Some are aggressively shouting fire, dumping excitatory chemicals. Others are whispering stay quiet, dumping inhibitory chemicals.
And the receiving cell just has to figure out who wins.
Exactly. The receiving cell body acts as a continuous biological calculator. It sums up all the yes votes and all the no votes millisecond by millisecond. Only when the excitatory signals overpower the inhibitory signals by a specific critical threshold does that neuron generate its own electrical domino wave and send the message down its own axon.
When you step back and look at the sheer scale of this voting system, it is dizzying. We already established there are 86 billion neurons, but the true source of your intelligence, your memories, and your personality isn't that raw cell count, is it?
No, it's not.
It's the number of those tiny gaps. It is the synapses.
The connection density is the true engine of human capability. The estimates suggest that the human brain contains roughly 100 trillion synapses.
100 trillion.
100 trillion chemical voting stations, constantly calculating, constantly summing up inputs. It's a number so large, it completely defies human comprehension.
I can't even picture a number that big.
Well, if you tried to count to 100 trillion, simply saying one number out loud every second, it would take you over 3 million years to finish.
Wow. And it's not even a static structure, which brings us perfectly to a concept that is so crucial to understand neuroplasticity.
A very important concept.
Because with 100 trillion connections constantly firing, voting, and summing, you might picture the brain as this fixed, rigid, unimaginably complex circuit board.
Right, like a computer.
Like the motherboard of a supercomputer that is soldered into place at birth and just runs its programs. But the brain is entirely different. It is constantly physically changing its own hardware based on those exact chemical and electrical signals.
And we really need to define neuroplasticity carefully here, because it is a term that has been thoroughly distorted by popular culture.
It really has. It has become the ultimate buzzword for the self-improvement and wellness industries. You see it on book covers, seminars, and life coaching blogs all the time.
Oh, everywhere. Rewire your brain for success.
Exactly. Rewire your brain for immense wealth, or unlock the neuroplasticity of eternal happiness. Let's strip away all of that marketing hype. What is the actual physical basis of neuroplasticity in the firm jelly of the brain?
In strict physical terms, plasticity simply means the brain alters its structural connections in response to experience and use.
So the connections physically change?
Yes. Remember those synapses, the tiny gaps where the chemical voting happens?
Yeah.
They're not fixed bridges. When two connected neurons are repeatedly activated together, when the first one fires and successfully contributes to making the second one fire, the physical and chemical connection between them strengthens.
The famous phrase is, neurons that fire together, wire together.
Exactly. On a physical level, the sending cell might start releasing more neurotransmitter ferries. The receiving cell might build more receptor docks to catch them. The actual physical shape of the dendrite can change, becoming thicker and more robust.
So the voting power of that specific connection increases.
Precisely. Conversely, if a connection is ignored or rarely used, that synapse weakens and can eventually be pruned away entirely. Sometimes, completely new synapses form.
So neuroplasticity is simply the physical manifestation of learning.
That is precisely what it is. Everything you know right now, every childhood memory you hold, every physical skill you have mastered, is encoded not as data on a hard drive, but as a specific physical pattern of connection strengths across millions of synapses.
But because of that self-help angle I mentioned earlier, I have to ask, does this mean there are no limits?
Limits to plasticity.
Yeah, like, can I just practice positive thinking, do some brain puzzles, and magically rewire my entire brain to become a concert pianist and a mathematical genius in my 40s?
This is where we have to firmly debunk the hype. Plasticity does not mean the brain is infinitely malleable.
So it's not just wet clay.
It is not wet clay that you can mold into whatever shape you desire simply through willpower. You cannot decide to relocate your visual processing center to your frontal lobe. You cannot fundamentally alter your baseline temperament just by wishing for a rewire.
So it's a biological mechanism, not magic.
It is heavily bounded. Plasticity is a very real, measurable physical process, but it is slow, highly energetically demanding, and bounded by biology. Furthermore, the degree of plasticity changes drastically over your lifespan.
Meaning it's different for kids and adults.
Completely different. The brain's ability to structurally change is at its absolute wild peak in childhood. Children go through what developmental biologists call critical periods.
What are those?
They're wide-open windows of time where the brain is incredibly receptive to profound structural change based on the environment. This is why a young child can absorb a complex new language with seemingly zero effort, perfectly mastering the phonemes, while an adult can study the same language for 10 years and still struggle with a thick accent.
The window just closes.
In adulthood, the biological window closes. The brain becomes much more constrained. You can still learn, obviously, but the physical changes are slower, require vastly more repetition, and are far more localized.
And there is a darker side to neuroplasticity that rarely makes it onto the self-help book covers. We are heavily conditioned to assume that plasticity is automatically a good thing.
Yes, that's a huge misconception.
It implies personal growth, resilience, and learning. But the reality is much more complicated.
We must remember that biology is entirely neutral. Plasticity is not inherently good or bad. It is simply the brain efficiently learning and solidifying a pattern. It does not possess a moral compass to judge whether that learned pattern is ultimately beneficial to your life.
What does that look like in practice? When is it harmful?
Some of the most profound examples of neuroplasticity in human behavior are addiction and chronic pain.
Addiction is a form of neuroplasticity.
Oh, absolutely. In the case of severe chemical addiction, the brain is ruthlessly and efficiently rewiring its reward pathways. It is strengthening the synapses that prioritize the seeking and consumption of a specific chemical substance, and simultaneously weakening the neural networks associated with other rewards, like social connection or long-term goals.
Wow, so the brain is learning exceptionally well.
It's just learning a pattern that destroys the organism.
And chronic pain works similarly.
Very similarly. In many cases of chronic pain, the initial tissue damage, say a lower back injury, has completely healed on a structural level. The discs and muscles are fine.
But it still hurts.
Exactly. Because during the injury, the nervous system repeatedly fired pain signals. Through neuroplasticity, the synapses in the pain pathways strengthened. They became incredibly efficient at firing. The nervous system essentially learned to amplify pain signals, and now it continues to fire them loudly long after the physical threat is gone.
That is neuroplasticity in action.
It is highly efficient biological learning, but it is deeply harmful to the individual.
That completely reframes the concept for me. It is just a mechanism of efficiency, for better or worse.
Exactly.
So we know how the connections change, and we know they form these massive networks of 100 trillion synapses. But where are these specific connections actually clustered? The brain isn't just a homogeneous bowl of jelly. It has specialized geography.
It's highly organized.
Let's take a tour of this real estate, working our way from the bottom up.
Geography is crucial, although we must always caveat that functions are highly interconnected. But broadly speaking, yes, there are distinct neighborhoods.
Starting at the bottom.
If we start right at the base of the skull, where the spinal cord enters the head, we find the brainstem.
This is essentially the brain's survival mode. This region handles the functions that you really do not want to be consciously responsible for managing.
You definitely don't want to have to think about them.
Right. It regulates your breathing rate, your heartbeat, your blood pressure, and your fundamental sleep-wake cycles.
It manages the absolute non-negotiable life support systems. Because these functions are so utterly essential for moment-to-moment survival, the brainstem is deeply protected. But it also means that physical damage to the brainstem, whether from a targeted stroke, a tumor, or traumatic swelling, is almost universally fatal.
Because it just stops the engine.
Exactly. If the brainstem goes offline, the basic rhythmic pacing that keeps the heart beating and the lungs expanding immediately ceases. You can survive massive damage to higher cortical areas and remain alive, but the brainstem is the biological foundation.
Moving just behind the brainstem, tucked underneath the main bulk of the brain at the back of your head, we have the cerebellum. And the statistics regarding the cerebellum are genuinely astounding.
They really are.
It is a relatively small volume of tissue. It looks like a little cauliflower hanging off the back, but it contains more than half of all the brain's 86 billion neurons.
The cellular density in the cerebellum is unlike anywhere else in the nervous system. The neurons there are packed incredibly tightly.
What does it do with all those neurons?
Functionally, the cerebellum is the supreme motor engine of the brain. It does not initiate the decision to move, that happens higher up, but it coordinates the movement.
Mhm.
It manages balance, posture, and crucially, it handles the microscopic precision of timing.
Is this why physical skills eventually become automatic? Like when you first sit down to learn a chord on the guitar, it is excruciating.
Oh, totally. You are consciously commanding every single finger.
Right, you're commanding every finger to move to a specific string, checking your hand visually, adjusting the pressure. But 10 years later, you don't even look at your hand. You just think G chord, and your fingers instantly fall into the perfect shape.
That is exactly the cerebellum at work. It takes those conscious, clunky, poorly timed motor commands from the cortex and through millions of repetitions, refines them.
It encodes the timing.
It encodes the perfect timing and sequence of muscle contractions into its densely packed networks. It allows you to execute highly complex physical actions, whether it's playing a guitar, typing on a keyboard, or just walking down a flight of stairs while drinking coffee, without dedicating conscious computational power to the mechanics of the movement.
Moving deeper inside the center of the brain, nestled above the brainstem, we find a collection of areas broadly referred to as the limbic structures.
The emotional center, broadly speaking.
Right. This is generally understood as the neighborhood where memory and emotion live. There are two major players here that we should define: the hippocampus and the amygdala.
Let's start with the hippocampus. It's an elegant, seahorse-shaped structure that is absolutely essential for the formation and consolidation of new long-term memories.
Does it store the memories like a hard drive?
No, it doesn't permanently store all your memories, but it acts as the necessary gateway. If your hippocampus is severely damaged, as was seen in famous neurological case studies, you would retain your intelligence, your personality, and your older childhood memories.
But you couldn't make new ones.
Exactly. You would be entirely unable to form a new conscious memory. You could have a conversation, leave the room, walk back in 5 minutes later, and have absolutely no recollection of the person you just spoke to.
That's terrifying. And sitting right adjacent to the hippocampus is the amygdala, which is famous for its role in emotion.
Specifically, the amygdala is heavily involved in processing emotional salience, which often manifests as fear or threat detection.
Like an alarm system.
It acts as an incredibly rapid, deeply ingrained biological alarm system. It is constantly scanning sensory input, flagging things in the environment that are emotionally significant or potentially dangerous, and triggering the physiological changes, like a racing heart or sweating, before your conscious brain even fully recognizes what the threat is.
And finally, we move outward to the wrapper. The part everyone visualizes when they think of a brain floating in a jar. The cerebral cortex.
The wrinkled part.
Yes, it is this deeply wrinkled, folded outer layer of tissue. I think everyone at some point has wondered why it looks so crumpled, like a walnut. And the physical explanation for this is just a brilliant packing solution by evolution.
It is a remarkable piece of biological engineering. The cortex is the seat of our highest level processing. It is where complex perception, abstract thought, and conscious awareness seem to be anchored.
So you want a lot of it.
Right. To increase an organism's computational power, evolution needed to add more cortical tissue. More tissue means more neurons and more synapses. However, hominids ran into a hard biological constraint.
The size of the human skull.
Exactly. A skull can only get so big before passing through the birth canal becomes a biological impossibility.
So you have a fixed, hard, spherical container, but you need to fit a massive sheet of tissue inside of it.
How do you maximize surface area within a restricted volume? You crumple it up.
It is exactly like trying to shove a massive winter sleeping bag into a tiny nylon stuffed sack.
That's a perfect analogy.
You can't just lay it flat. You have to aggressively fold it and compress it, creating all these deep grooves and ridges just to maximize the surface area that fits inside the limited space.
That is precisely what the wrinkles, the sulci and gyri of the brain achieve. If you were to somehow extract the human cerebral cortex and iron it out completely flat on a table, it would be roughly the size of an extra-large pizza or, a large newspaper page.
A pizza inside your skull.
All those deep wrinkles allow that massive sheet of computational power to be jammed into the tight confines of the skull.
And this vast pizza-sized sheet of cortex is broadly divided into 4 major territories, or lobes, on each hemisphere of the brain. Let's map these out slowly so you, sitting there listening, can actually visualize where these functions are sitting on your own head.
Okay, let's trace it.
Starting at the very back of the skull, just above the neck, you have the occipital lobe.
The occipital lobe is almost entirely dedicated to the massively complex task of vision. It takes the raw electrical signals sent from the retinas of your eyes and begins the arduous process of decoding them.
Like finding the edges of objects.
Exactly. It finds edges, it detects motion, it recognizes color, and it begins to assemble those fragments into the cohesive visual world you experience.
Moving around to the sides of the head, roughly in the area right around and behind your ears are the temporal lobes.
The temporal lobes are the primary centers for processing auditory information hearing. Crucially, on the left side, for most people, this is where language comprehension is heavily anchored.
And they sit right next to the hippocampus, right?
Yes, the temporal lobe is deeply connected to the hippocampus, making it a critical region for integrating those auditory and visual inputs into complex, retrievable memories.
Then, moving up to the top of the head, sitting like a crown, is the parietal lobe.
The parietal lobe is the grand integrator of sensory information. It processes the sense of touch, temperature, and pain from across your body.
And spatial awareness, too.
Yes, profound spatial awareness. It constantly calculates where your limbs are in three-dimensional space without you having to look at them, and it integrates that map of your body with the visual map of the world around you so you can navigate without constantly crashing into walls.
And finally, we reach the massive section occupying the entire front of the skull, sitting right behind your forehead. The frontal lobe.
The frontal lobe is arguably the region that makes human behavior so distinctively human.
It doesn't do raw sensory input, right?
No. Instead, it is the executive command center. It handles complex, multi-step planning. It weighs the outcomes of difficult decisions. It originates the command for voluntary movement. And, critically, it governs the crucial social skills of impulse control and emotional regulation.
And the frontal lobe deserves a very deep dive here because of its unique developmental timeline. We think of organs generally growing and maturing together, but the frontal lobe is on a massive delay.
It is the absolute last region of the brain to fully biologically mature.
How late are we talking?
Well, while your sensory regions are fully locked in very early in life, the physical wiring and the insulating myelin sheaths of the frontal lobe continue to actively develop and solidify well past adolescence, typically continuing into a person's mid-20s.
Think about the profound, real-world implications of that biological fact. The area of the brain strictly responsible for overriding impulses, for foreseeing long-term consequences of dangerous actions, and for regulating erratic behavior, is essentially a construction zone with wet paint and scaffolding throughout the entirety of the teenage years.
It provides an enormous amount of biological context for adolescent behavior. Teenagers have highly active emotional centers. Their amygdalas are firing on all cylinders. But the executive braking system in the frontal lobe is not yet fully installed.
And this biological reality has actively changed how society operates, specifically within the legal system.
It has forced a major reckoning in juvenile justice, yes.
Because if the hardware isn't there,
Exactly. If the biological hardware required for mature impulse control and risk assessment is simply not yet physically mature in a 19-year-old, it forces courts and society to ask highly difficult questions about ultimate culpability.
it shifts the conversation toward rehabilitation rather than pure punishment for offenders under the age of 25, because their brain architecture is quite literally not finished developing.
Precisely.
When we talk about the frontal lobe and personality, it is impossible not to discuss the most famous cautionary tale in the history of neuroscience. The story of Phineas Gage.
Oh, everyone has heard of Phineas Gage.
It's a story almost everyone has heard a version of. The year is 1848. Gage is a young, capable railway foreman working in Vermont. He is packing explosive powder into a rock hole using a heavy iron tamping rod.
A very dangerous job.
Very. A spark ignites the powder prematurely, and the explosion shoots this massive iron rod over a yard long and an inch thick, straight up through his left cheek behind his eye, and violently out the top of his skull, completely destroying a massive chunk of his frontal lobe.
And against all medical odds for the 19th century, he survives.
His survival alone made it a medical marvel. But the reason Phineas Gage became a foundational pillar in every psychology textbook is the reported aftermath of the injury.
Right. The classic dramatic narrative states that Gage survived, but his personality was entirely obliterated.
He reportedly went from being a polite, hardworking, highly respected foreman to a completely impulsive, profane, animalistic person who could not hold a job or conform to social norms. The famous quote from his physician was, "Gage was no longer Gage."
It seemed to perfectly neatly prove that the frontal lobe was the sole seat of human morality and restraint.
But the reality of Phineas Gage is vastly more complicated than the textbook myth, isn't it? The historical narrative has been aggressively streamlined.
It is one of the clearest examples of scientific folklore in modern history. Did Gage suffer catastrophic frontal lobe damage?
Yes.
Did he experience severe behavioral changes immediately following the trauma? Absolutely.
So what's the myth part?
Over the ensuing decades, textbook authors heavily embellished his symptoms to make a cleaner, neater pedagogical point about the strict localization of brain function. They turn him into a one-dimensional monster.
What does the actual historical record show about the rest of his life?
The historical records, carefully unearthed later, suggest that Gage actually experienced a profound degree of social recovery. Later in his life, he traveled to Chile, where he successfully held down a demanding job driving a stagecoach for years.
Driving a stagecoach in Chile, that's not a job for someone who is completely impulsive.
Exactly. Driving a stagecoach with a team of horses requires immense multi-step planning, sustained focus, complex motor control, and the ability to interact with passengers. A completely impulsive, uninhibited person could never maintain that job.
So his remaining brain tissue, perhaps utilizing that neuroplasticity we discussed earlier, found workarounds. It compensated for the damage.
That's the most likely explanation. The true lesson of Phineas Gage is not that the manners. The lesson is that the brain is highly interconnected, incredibly resilient, and capable of profound compensation.
It's a warning not to oversimplify things.
Exactly. We must always be wary of incredibly neat, overly dramatic narratives that perfectly support a simplified theory.
And that transition into scientific folklore brings us to a vital part of our mission today. Because the brain is so wildly complex, and because we all possess one, misinformation about it is unusually persistent. It sticks in the cultural consciousness and refuses to die. We need to clear out some of these cobwebs. Let's methodically break down and bust some of the most persistent brain myths that are still actively circulating.
I am more than happy to do this. The sheer volume of biological misinformation in popular culture is staggering.
Let's start with myth number 1, the absolute classic, undisputed king of brain myths, the 10% myth. I cannot even count how many blockbuster movie plots rely entirely on this premise.
Oh, it's so common.
The protagonist takes an experimental pill or gets exposed to radiation, and suddenly the scientist says, we only use 10% of our brains, but he just unlocked the other 90%. And suddenly the character has telekinesis, can predict the stock market, and learns conversational Mandarin in 5 minutes.
It is a fantastic hook for a science fiction movie. It taps into our deep-seated desire for untapped potential. But biologically, physiologically, and neurologically, it is entirely unequivocally false. We use 100% of our brain.
If someone asks for the concrete proof, how do we definitively know there is no hidden 90%?
We can prove it from multiple independent scientific angles. First, we have modern functional brain imaging, like fMRI and P scans.
What do those show?
Whether a subject is performing a highly complex calculus equation, reading a novel, or simply sleeping soundly, functional imaging shows waves of electrical and metabolic activity sweeping constantly throughout the entire mass of the brain. There are no dark, dormant, completely silent biological voids just sitting there waiting for a magic pill to activate them.
And the second angle ties right back to our opening discussion about energy, right?
Precisely. The metabolic cost absolutely destroys the 10% myth.
Because it takes 20% of our calories.
Right. Maintaining living neural tissue is phenomenally expensive. If 90% of that tissue were completely idle, evolution would have ruthlessly eliminated it millions of years ago.
Right.
It would be evolutionary suicide for a species to carry around 3 extra pounds of highly demanding, calorie-burning tissue if it served no active function.
That makes total sense.
Furthermore, in clinical neurology, we observe that damage to almost any area of the brain, even a tiny stroke in an obscure region, results in some specific measurable deficit. There is no spare 90% that you can comfortably damage without consequence. Every region has a job.
Consider myth number 1 entirely busted. Moving on to myth number 2. Left brain versus right brain personalities. You see this everywhere in corporate team building exercises and online personality quizzes.
Those quizzes are everywhere.
The premise is that you are either a logical, analytical, math-driven left-brained person, or you are a creative, artistic, free-flowing right-brained person.
This myth is particularly tricky to eradicate because, unlike the 10% myth, it is built on a very tiny actual grain of neuroscientific truth. But that truth is then wildly, inappropriately extrapolated.
What is that initial grain of truth?
The grain of truth is a concept called lateralization. The two hemispheres of the brain do, in fact, show some specialization for certain tasks.
Like what?
For example, in the vast majority of the population, the heavy lifting for fine language processing, grammar, vocabulary, speech production is strongly lateralized to the left hemisphere. Conversely, certain complex spatial integration functions, or recognizing faces, lean more heavily on the processing power of the right hemisphere.
So if language and math lean left, why does it remain a myth that I am a left-brained person?
Because lateralization of a specific sensory or motor function does not translate into a unilateral global personality type. There is no such thing as a unilaterally logical half of a brain and a unilaterally creative half.
They don't work independently.
The brain does not silo complex behaviors. Whenever you perform essentially any high-level task, whether you are painting a landscape, solving a differential equation, or trying to understand a sarcastic joke, both hemispheres are lighting up simultaneously and working together in constant concert.
And they are physically connected. They aren't two separate computers.
Exactly. They are bridged by a massive, dense band of white matter called the corpus callosum. This structure is composed of roughly 200 million individual nerve fibers.
200 million.
Yes. It acts as a massive biological broadband cable. Those 200 million fibers are constantly, frantically exchanging billions of bits of information back and forth between the hemispheres every second. The hemispheres are in constant dialogue. You are not a left-brained person or a right-brained person. You are a wholly integrated, whole-brained person.
Yeah. Let's move to myth number 3. And this one is so deeply embedded in our global education system that it feels almost heretical to question it. Learning styles.
Yes, this is a big one.
It's the deeply held idea that students are inherently either visual learners, auditory learners, or kinesthetic learners, and that teachers must tailor their instruction to match a student's specific style for them to learn effectively.
This is perhaps one of the most frustrating myths for cognitive scientists and educational psychologists to combat. It has immense intuitive appeal. It feels correct.
But is it?
When the theory is rigorously scientifically tested in controlled environments and has been tested repeatedly over decades, the core claim consistently fails.
Wait, I really need to drill down on this because I personally feel a strong conviction that I learn better when I see a clear diagram rather than just listening to a lecture. How can it be a myth?
The key is distinguishing between a subjective preference and a measurable educational outcome. People absolutely have strong preferences.
Okay, so I prefer it.
Right. You might deeply prefer to look at a diagram. Someone else might prefer to listen to an audiobook. But preference is not the same as a biological constraint on learning.
What happens when they test it?
When researchers actually run the empirical experiments, when they take a group of self-identified visual learners and randomly assign half of them to learn a subject via text and the other half to learn via diagrams, the outcomes, the actual test scores assessing their comprehension, do not show a statistically significant difference based on matching their preferred style.
So matching the teaching method to the self-reported style doesn't actually boost the learning.
It simply does not. What cognitive science actually tells us is that the best way to teach something depends almost entirely on the material being taught, not a fixed neurological style of the student.
That makes a lot of sense.
If you are teaching the geography of South America, everyone needs a visual map, whether they think they are an auditory learner or not. If you are teaching the cadence of a Shakespearean sonnet, everyone needs to hear it spoken out loud.
You can't just read about the rhythm.
Right. Furthermore, we know that learning is vastly improved by active recall, forcing the brain to retrieve information, rather than passively absorbing it in your preferred medium. The learning styles myth wastes an immense amount of educational resources trying to categorize students into fixed boxes that biologically do not exist.
That is going to fundamentally shock a lot of listeners.
Yeah.
Okay, our final myth to bust, myth number 4, the fMRI glowing dot hype.
The glowing dots.
This addresses those incredibly flashy news headlines we see seemingly every week. Scientists find the exact brain region for love, or researchers locate the biological center for conservative political thought. And the article is always accompanied by a picture of a gray brain with one brightly glowing orange dot in the middle.
We most actively caution everyone to view those specific headlines with an extreme degree of skepticism.
Why? Isn't the machine accurate?
Functional MRI, or fMRI, is an incredibly valuable, sophisticated tool for neuroscience, but its findings are constantly oversimplified and exaggerated by the popular press. To understand why, it is vital to understand what an fMRI machine is actually measuring.
What is it measuring?
It does not directly measure neural activity. It cannot see neurons firing action potentials.
If it isn't seeing the electricity, what is the machine actually detecting?
It measures blood oxygenation. It operates on a principle called the BOLD-D, signal blood oxygen level dependent.
How does that relate to thinking?
The theory is sound. When a specific brain region is working incredibly hard, those sodium-potassium pumps are burning energy, so that region needs more oxygen. The circulatory system responds by sending more oxygenated blood to that specific area.
Okay, I followed.
The massive magnets in the fMRI can detect the difference in magnetic resonance between oxygen-rich and oxygen-poor blood.
So the glowing orange dot is just showing where the blood is flowing.
Exactly. But blood flow is a delayed indirect proxy for thought. It operates on a massive delay of several seconds. Neural activity, those electrical dominoes, happens in milliseconds.
That's way behind.
Therefore, fMRI has very poor temporal resolution. It cannot capture the split-second, incredibly complex dialogue happening across the entire brain. It just captures a blurry, delayed snapshot of blood pooling.
And this brings us to one of the most hilarious yet profoundly important cautionary tales in the history of modern neuroscience, the dead salmon study.
It is absolute legend in the field and a masterclass in scientific skepticism.
What exactly did they do?
A group of researchers wanted to demonstrate to their colleagues the massive statistical pitfalls inherent in analyzing fMRI data. An fMRI scan generates an enormous, almost unmanageable amount of data. It mathematically splits the 3D space of the brain into tens of thousands of tiny, three-dimensional pixels called voxels.
So you have tens of thousands of data points for every single scan.
Precisely. And when you run a standard statistical analysis looking for fluctuations across tens of thousands of voxels just by pure mathematical chance, random background noise will occasionally clump together and look like a genuine signal.
If you look at enough static, you'll see a shape.
Exactly. If you don't aggressively correct your math for multiple comparisons, you will find false positives everywhere.
So to prove this point, what did this research team do?
They went to a local fish market and purchased a mature Atlantic salmon, a deceased salmon.
Okay.
They placed the dead salmon into the fMRI scanner. They then ran a standard experimental protocol. They showed the dead salmon a series of photographs depicting human individuals engaged in various social situations, and they explicitly asked the salmon to determine what emotion the humans in the photos were experiencing.
I know this is serious science, but the mental image of a dead fish being asked to evaluate human emotions inside a multi-million-dollar medical scanner is incredible. And this was a real peer-reviewed paper.
It was a very real, highly influential paper. Because when they ran these standard, uncorrected statistical analysis that many researchers around the world were routinely using at the time, the scan showed an apparently significant cluster of brain activity in the dead salmon's brain cavity precisely while it was viewing the photographs.
The machine found a glowing orange dot of empathy in a dead fish.
Because of random, uncorrected statistical noise. It brilliantly and undeniably highlighted the vital need for rigorous statistical correction in a way that a dry mathematical methods paper never could have achieved.
So fMRI isn't useless, it's just easily misinterpreted.
Exactly. The point of the salmon is not that all fMRI is useless. Properly corrected fMRI research is eliminating. But when you see a headline claiming scientists found the exact isolated region for love, remember the salmon. Highly complex human behaviors like love, political affiliation, or moral decision-making are almost never localized to one single glowing dot. They are the emergent result of massively distributed processing across those 100 trillion synapses.
Which brings us to the final and, frankly, the most philosophically staggering section of our deep dive today. We call it the ultimate mystery. We have mapped the physical anatomy of the firm jelly. We traced the electrical dominoes and the chemical fairies. We busted the myths of left brains and learning styles. We can explain a staggering, triumphant amount about how the brain mechanically processes information.
The scientific progress over the last century is genuinely breathtaking. We can literally trace a visual signal step by step.
All the way through.
Yes, we can track photons hitting the retina, watch the electrical conversion, follow the signal down the optic nerve, to the thalamus, to the back of the occipital lobe. We can observe processing stages that extract geometric edges, then detect motion, then integrate color, and finally recognize a specific human face.
That's incredible.
We can implant electrodes and watch memories physically consolidate during REM sleep. We can even hook a person up to a scanner and, by observing motor cortex preparation, accurately predict that they're going to push a button seconds before they consciously report making the decision to push it.
But despite all of that incredible granular mechanical progress, we inevitably hit a massive glaring wall. And this wall is what the philosopher David Chalmers famously framed as the easy problems versus the hard problem of consciousness.
And we must clarify immediately that when Chalmers calls them the easy problems, he is being quite ironic.
Because they aren't easy at all.
The easy problems are actually enormously difficult, Nobel Prize-winning questions about biological mechanics. They are questions about how perception works, how the brain sustains attention, how memory is molecularly stored, and how motor behavior is generated.
But they're categorized as easy because they are fundamentally just matters of function, right? We
Yeah.
know what the biological puzzle pieces look like. We just have to dedicate enough decades and supercomputers to figuring out exactly how they snap together.
Exactly. They are ultimately complex engineering problems. Given enough time, technology, and funding, we are making steady, observable progress on them. We are solving the mechanics. But the hard problem is in a completely different, almost unapproachable category.
What is the hard problem?
The hard problem asks a question that neuroscience cannot currently answer. Why is any of this highly complex biological function accompanied by a subjective, internal experience?
Why does it feel like anything at all? If we go back to the visual processing example, I can entirely understand the mechanical chain reaction of a 700 nanometer wavelength of light hitting my eye, generating an electrical domino wave, sending glutamate ferries across a synapse, and lighting up a region in my occipital cortex.
You understand the mechanics of the data transfer.
Right. But why does that cold, dark, silent data processing suddenly and magically result in the vibrant, undeniable, subjective, internal feeling of seeing the color red?
Why is there something that it is like to be you? Why doesn't all this brilliant biological processing just happen entirely in the dark without awareness, exactly like a silicon computer processing a complex line of code?
Like a camera. A digital camera can detect red light perfectly, measure it, and categorize it, but we do not believe the digital camera experiences the redness.
It has no inner life. So how does the wet, firm jelly of the brain generate an inner life?
So what are the theories? How do scientists and philosophers even begin to approach a solution to the hard problem?
It is the frontier of human thought, and there are several competing theories, but it is vital to note that there is absolutely zero scientific consensus. There are two major frameworks that often lead the discussion.
That's the first one.
The first is called global workspace theory. This framework proposes that consciousness acts essentially like a theatrical spotlight on a dark stage. It theorizes that consciousness arises when a specific piece of information is broadcast widely across the brain's networks, becoming simultaneously available to many different cognitive systems at once. Your memory system, your verbal reporting system, your motor action system.
So when everyone gets the memo, you feel it.
Basically, when the information enters that global workspace, you become subjectively conscious of it.
Okay, so in that view, consciousness is just a state of widespread internal broadcasting of data. What is the other major approach?
The other major framework is called integrated information theory, or IIT. This is a much more abstract, mathematically driven approach.
How does it work?
It attempts to literally quantify consciousness. It proposes that consciousness is a fundamental property of complex systems, and it arises based on how densely integrated a system's information is. The more integrated the network, the more the parts mutually influence each other in a way that cannot be reduced to individual components, the higher its mathematical score of consciousness, a value they denote with the Greek letter phi.
But neither of these theories has definitively won out or solved the mystery.
Not even close. Both theories have highly passionate advocates and incredibly fierce, serious critics.
What do the critics say?
Global workspace theory might elegantly explain the mechanism of how we access information, but critics forcefully argue it still doesn't explain the subjective feeling of the information. It just explains the broadcasting.
And the math one.
Integrated information theory provides elegant, complex math, but its philosophical implications, which suggest that even highly integrated inanimate objects might possess a tiny fraction of consciousness, are highly controversial.
That is wild.
The deeply humbling truth is that we do not even have an agreed-upon scientific framework for what a final, satisfying answer to the hard problem would even look like.
It remains a profound, beautiful mystery. So as we finally wrap up this expansive deep dive, let's take a breath and look at the grand summary of what is happening inside your head at this exact second.
It's quite a lot.
You possess 86 billion microscopic workers. They are frantically communicating across a dizzying network of roughly 100 trillion chemical and electrical synapses. This vast, dense, metabolically expensive network is meticulously organized into specialized but heavily interconnected regions, from the deep survival-focused brainstem all the way up to the crumpled, pizza-sized wrapper of the cortex.
And this firm jelly is continuously, physically reshaping its own structural connections every single time you learn a fact, practice a chord, or experience something new.
And from this incredibly complex, wet biological machinery, we somehow derive absolutely everything that matters to human existence. We get the vivid colors of vision, the complexity of spoken language, and the capacity for multi-decade planning.
We get the profound, subjective, gut-wrenching experiences of love, joy, and grief.
And perhaps most astoundingly of all, from this 3-pound mass of matter arises a mind that possesses the astonishing capacity to turn inward and investigate itself.
Which is truly poetic when you think about it.
But that core mystery, the heart problem, remains untouched. Why does it feel like anything from the inside? I want to leave you, our listener, with a final provocative thought to mull over today as you go back to your coffee, your commute, or your laundry. It builds right on the edge of what we just discussed.
It's a great question.
If consciousness is fundamentally a mystery of why highly complex function produces this subjective experience, well, could we ever truly know if a non-biological system eventually achieves it? Imagine we build a highly complex, deeply integrated artificial intelligence system, a machine architecture that perfectly mathematically mimics the exact signaling and the 100 trillion connections of our own brain.
A perfect silicon replica.
Exactly. If it processes information with the exact same integrated complexity, does that silicon machine suddenly start feeling something from the inside too? Or is the subjective magic of feeling uniquely and exclusively tied to the biology of our living jelly? It is a question that pushes the boundaries of neuroscience right into the looming future of artificial intelligence. Thank you so much for joining us on this deep dive. Keep wondering about that incredible, complex universe sitting quietly right inside your own head.